We have a fill-in host right now. I'm gonna take a 30-minute break and give Felix Fernandez-Penny, an intern here at BTI, who has been here for a while, right? Yeah, a couple years at least.
5 now.
5 years. 5 years, and he was an intern in the Jander lab, and now he's one of our communications interns, and probably knows the building better than I do. So I want to give him a chance to host a segment here with Ning, and they're going to talk all about Ning's research at the Martin Lab. So take it away, Felix.
Thank you, Keith. Hi, so Ning joined us for the last segment, but if you weren't able to tune in then, I'll just go ahead and have you kind of reintroduce yourself and tell us a little bit about your professional background and what brought you here to BTI.
Okay. I'm Ning. I'm now a postdoc in the Martin Lab. So I have been in the lab for 1 year. So before that, I got my bachelor and master degree in China. So as an undergraduate, I studied landscape architecture.
Oh wow!
And then I transferred to silviculture, which I focused on the tissue culture and the transformation in bamboo.
Okay.
Yeah. After that, I went to the Ohio State University for my PhD.
The Ohio State.
Yeah. So I further studied soybean transformation and promoter analysis. So I finished my PhD last year. And then at that
Congratulations.
time, at that time, the Martin Lab were looking for a postdoc who can take the lead in the CRISPR project in the lab. So because I also did some CRISPR project during my PhD, which I used CRISPR to do precise transcription modulation. So I'm very interested in those CRISPR projects, and also I know BTI was a very prestigious institute for plant science.
Yeah.
So I applied for the job, and then I joined the lab last November.
It worked out, I'm guessing you're here. So that's great. So you tossed around the word CRISPR a little bit, and I'm a little bit familiar with that just based on kind of what I've heard throughout the BTI community and whatnot, but would you mind touching a little bit on kind of what the CRISPR platform is and its use, and maybe your involvement with it?
Okay. Yeah, CRISPR, I think the full name of CRISPR is clustered regularly interspaced short palindromic repeat. It was first discovered in most bacteria and archaeas. So it's a kind of naturally existing immune system for them to recognize foreign DNAs like virus or plasmid. So I think it was first described in 1987 by a Japanese scientist. But it was not until 20 years later scientists began to realize that it was a great tool to use for genome editing. So I think in the past 5 or 6 years, it has achieved a great improvement in CRISPR. So thousands of papers have been published in different areas using these tools to do DNA.
All within plants usually, or can CRISPR system be widely applied?
Widely applied, not only in plants. I think in animals it's really more.
It's really universal.
Yeah, universal. It's very easy for scientists to use because you only need to design about a guide which is about 20 base pair. It's unique to the genome. And then the guide when it expressed, co-expressed with a Cas9 proteins in the cell, the guide scaffold can bind to the Cas9 proteins and then guide those protein complex to the targeted region. And then it can induce a double.
Come in right exactly on.
And then at that region it can induce a double-strand break. So the cell itself can fix the break due to the endogenous mechanisms. And so during this process, mutations can be induced, and also other modifications can occur based on the scientist's objectives.
Wow.
So it's it really have multiple applications, like I said, in biological research, in medicine, and in agriculture. So I think the most common use in biological research is to generate those gene knockout. So those mutations will be a great resource to study the loss of function of those genes.
And better classify genes and kind of identify their role.
Yeah, that's true. And also other applications like DNA modification, like DNA labeling or RNA targeting, and even transcriptional modulation. So all kinds of different applications.
That's great.
Yeah.
It sounds like a dream come true for the gene editing science
Yeah,
and whatnot,
yeah.
for transformation in
Yeah.
general. So I suppose turning a little bit to your research, how are you using CRISPR, and how is CRISPR kind of helping you meet the objectives of your research project?
Okay. Like I said, in my lab mainly studies how bacteria infect plant and how plant defend themselves under those attacks. So we use tomato as a model system to study bacterial disease caused by a pseudomonas. So like in my project, because like our lab has already identified many potential immunity-associated genes, but before the advent of CRISPR, we can only use nicotinamide or
certain model plants.
Yeah, it's like like a different system to study those genes, but it may not accurately reflect the gene function, right? So now we have CRISPR. So my project is focused on generate those mutant lines that knock out those genes in those in tomato. So we
in
aim,
tomato.
yeah, in tomato. So we aim. Our aim is to generate about 250 different mutant lines.
Wow,
so that will be a great resource for us to study the gene of the function in tomatoes, and then we can better understand how this gene is involved in pathogen response. Yeah. So I think it will be very good for the.
That's great.
Yeah,
so you touched on it briefly just a little bit here, but kind of implications for the wider scientific community. And when we spoke earlier, you mentioned some unique collaborations that you have with other labs and other members of the kind of plant science and scientific community. Could you touch a little bit on those?
Yeah. And now we are our collaborator is the Phi Lab. So they are a bioinformatics lab.
Right.
So
they were just with us earlier this morning, if you caught their segment.
Oh, yeah. So they help us develop a tomato CRISPR database.
Okay.
So in this database, we will input all our information about our mutant lines, including the guideline we use, the mutation types, and also the different generations, and also even the immunity response of those plants. And the morphology of those plants. So finally, I think we will make those database public, so all the scientists can get access to this database.
And draw from that.
Yeah, and they can even request seed from us if they are interested in those mutants. And also, we think we can also invite other scientists to use those database to submit all their information about their own mutant, generated by CRISPR. So I think this will be a very cool
resource
for our scientists, especially for scientists in our area.
Right.
Yeah.
And I imagine there's just such a huge amount of data there that compiling that all together could be such an asset, for various research projects.
Yeah, that's true. Yeah.
That's great.
Yeah.
Wow.
You talk about the collaborator, right?
Yeah.
Yeah, I think I will have another collaborator also in BTI. It's the Joyce Lab.
Oh, okay.
So their lab is a transformation lab. So now they do almost all our transformation by their lab. So that was really important. That's really important for us because if we don't get the mutant lines, we can't do any analysis to see the gene function like that. So we think collaboration between different labs was really important. It not only saves your time and energy, it also makes your project go smoothly and fast, right?
Absolutely.
Yeah.
So you were talking a little bit earlier about kind of the more widespread implications of some of this work on pathogen resistance and whatnot. And I wonder if you could talk about some of kind of the most pressing issues that face the agricultural community right now, kind of in terms of where the need is, how these projects can best support, whether or not it's an international issue, if it's something that's more localized, kind of the broader implications of this work.
You mean my work or CRISPR?
I mean, feel free to touch on either your work specifically, CRISPR as a platform.
I think the most like challenge for CRISPR now is like the first is the off-target. Well, the one thing is about off-target TC questions.
Okay.
So I think they also like the society also try to solve this problem. So they try to use different Cas proteins. It's not only Cas9. Cas9 is just one protein.
Right.
So they try to find different varieties of those proteins to see if they have better specificity and other stuff to solve this problem. Other things I think is this is also trying to identify different Cas proteins is also another like solution to make the proteins better to go into the cells, because the Cas9 is really big proteins. So they want to find some varieties smaller.
Right.
So for especially in cells, like in human cells, for those research they like to have small proteins. So it's better for them to go into plant cells.
Right.
I think other stuff is like the because gene knockout is really easy now, but the other issue like gene replacement is still a challenge. It can achieve, so some it's still possible, but it's at a very low efficiency. So this is I think is another thing the scientists want to make it more efficient.
Yeah. So you talked about a little bit about kind of the timelines and the ease of transformation and whatnot. For those of us who aren't so familiar with transformation and whatnot, could you just go over what kind of a typical timeline for creating a new transform and gene knockout, kind of all the research and analysis that goes into it, and then the wet lab work, just a little bit of kind of the timeline of how you might get from point A to having a knockout.
Okay, so now like for making the contract is really easy. It may take like 1 week, even 2 weeks to make multiple.
Right. You just have to wait for it to be
yeah,
sent over.
Yeah, it's really easy because you can pull out all your contracts even together and to do one transformation ligation. So it's easy. But for transit expression is also very short. So it's about 2 weeks. You can get a transit expression even in benzimiana or other, in tomato.
And the expression usually lasts about 2 weeks?
No, it's like the peak expression is about 3 to 5 days.
Oh, okay.
But I mean the whole process, because you need to prepare all the agars and also
all the
other stuff.
materials.
Yeah. So because we use transit expression to test the guide-ion efficiency, so you can test them. But for stable expression, it really takes a long time.
Right.
I take an example the tomato. So I think before, like from the time we send the contract to the lab, to John's lab, and to the time we got the plant seedlings, it takes about 4 months. I think.
4 months.
4
Wow,
months.
okay.
But we need to transfer the seedlings to the soil, and then we after they get the fruit, it will take another 3 to 4 months.
Wow,
so it's
okay.
a long time.
So it's quite a process.
Yeah, but also this is only the first generation of the mutant. Like the first generation, we need to go through a next generation to get the homozygous mutation. Also,
so it's fixed. So
yeah, because in the first generation, most of the time is a heterozygous mutation. So not all the gene alleles are mutant, only part of them. So in the next generation,
you're never sure whether or not you're going to have the mutant.
Yeah, that's true. They will segregate,
okay.
right? So we will get the one that has all the alleles are mutants. And ideally, we like to get rid of the transgenes, like Cas9, something, like that. So
you just have what you're interested in.
Yeah, so we will usually go to another or even another 1, even 2 generations to get the ideal mutant. So it takes another half a year or something
Wow.
even more.
So quite a long
yeah, it's a
involved
long
process.
time. It's a long time
Okay,
to
yeah.
get the idea.
I mean,
Yeah.
it sounds like this technology really just affords, so much potential in all fields of fundamental science research, and it's been really interesting hearing a little bit about your work and kind of the broader implications of the CRISPR platform and the like. So I just want to thank you again so much for joining us for 2 sessions in a row today as part of BTI Live at BTI Giving Tuesday. Ning, thank you so much for being here.
Yeah, you're welcome.